We're about to start panel 2, Agents for Chemical Reactivity. My name is Dunstan Macauley. I'm the moderator for this panel. Our panelists for this panel are Dr. Jonathan Williams with the Max Planck Institute for Chemistry, Dr. Rachel Adams, Indoor Air Quality Section, Environmental Health Lab of the California Department of Health, and research scientist at the University of California at Berkeley, and Dr. Barbara Turpin, who is a professor and chair of environmental sciences and engineering at the University of North Carolina at Chapel Hill. Our first presenter is Dr. Jonathan Williams.
Thank you very much, and good morning. Yes, today I'm going to be talking now about people chemistry. And just to set that a little bit in context, we heard this morning from Alan and from Richard and from Nina that the indoor environment contains a sort of huge array of complex and very diverse sources. So, for example, cooking, and cleaning, and also the emissions from these various furnishings. And what that means is that trying to get a general impression of the indoor chemistry of the home is very complicated because it can vary very much from one place to the next. So now what we're going to do is we're going to turn our attention to the one common element in all living spaces, and that is the person. Okay? And we're going to ask ourselves the question, what do people emit into this cocktail of chemicals on their breath or on their skin? Or in other words, what are you right now and I emitting into this auditorium for everyone else to breathe? Okay, so we're going to look at that. And here's some examples of people doing exactly that. Okay. So my group got into this line of inquiry through some very unusual measurements that we did in our local movie theater. And it turns out that a movie theater is an excellent piece of scientific equipment to measure the emissions from people. The reason for that is you get a large group of people, so you get an average sample of 250 folks watching a movie, and while they're watching the movie, you pass fresh air from outside under the seats and out through the people and then out through the ceiling, out through a ventilation shaft. And what that means is that anything that the people are emitting, it's basically a box full of people that's flushed with air, anything that they emit can be measured downstream using some of the highly sensitive equipment that Alan was also demonstrating earlier on. So we were measuring CO2, as you know, major gas in your breath, and also a huge number, so several hundred of these volatile organic compounds, some of which you saw again earlier on today. And so what we did was what we found, because our instrumentation is rather sensitive and measures continuously, you can pick up on very subtle changes in the gas phase composition that's caused by people reacting to various scenes in the films. Okay? And this film, as you can probably identify from the peaks, right, is The Hunger Games 2, and you see that when this film is shown to multiple audiences multiple times, there's a certain reproducibility. So you start to see peaks happening in the same places. That's for CO2. So you can see people getting excited or not excited. We also, because of the hundreds of chemicals that we had at hand, we were also able to pick out certain scenes were linked to certain VOCs. So there seemed to be an expression in the air. We're sort of reading emotions, if you like, out of the air by taking these sort of measurements. As a kind of spin-off to that, we actually used this database of films to actually be able to calculate the recommended age classification for unknown films too. So that's something we've also published if that's of interest to you. But that's kind of just the introduction of how we got into the indoor thing. One thing that we, of course, became aware of through these studies was that the real-world emissions from people are really rather large. As Alan said too, probably the main source in this room are ourselves. Okay? And in the real world, we were able to capture measurements of these siloxanes from cosmetic products, or from external. We were able to measure breath compounds like isoprene, and also ethanol, of course. If you've drunk beer or so, you're emitting it also into the environment too. So it's a real-world situation. It's very high rates, and so we thought, okay, this is an important compound for indoor chemistry. We need to look at it under more controlled conditions and to really find out what a human being just living in a space produces for chemistry. And to do that, we took it out of the cinema and went to the more controlled conditions of a climate chamber, which is located in Denmark, run by the Danish Technical University. And so what we did was we basically ran an experiment with 4 people in a chamber under various conditions, which I'll describe to you in a minute. And that was part of this project, ICHEA, the Indoor Chemical Human Emissions and Reactivity Project, a Sloan-funded project. And what you see here is actually a rather unusual case. Normally, we would simply take 4 happy, healthy Danish students, put them in the climate chamber, and ask them to play with their mobile telephones for a few hours while we measured them. And in this experiment that you're seeing here, it's a special case. What we did there is we applied masks to the students so that we could separate what was coming from the skin and what was coming from the breath. Okay, so we could get like a separate measurement of both. So on the right, you can see the various measurement technologies we applied to this. And you can see they're all in sort of rather strange shaped racks. And these are flight racks because these measurement systems normally fly on a high-altitude aircraft and measure the very low concentrations that you get at the top of the atmosphere or in the stratosphere. And what we had was like proton transfer reaction mass spectrometers. We had a custom-made fast GCMS. We had an interesting new measurement to indoor chemistry called OH reactivity. I'll show you some results from that in a second. And on top of that, so kind of more standard stuff, ammonia, ozone, NO, CO2, methane, and of course some ultrafine particles. Now, of course, if we got a chamber, we can control the conditions. So we can run a series of experiments, each time with 4 happy, healthy students with fragrance-free washed clothes. We put them in there, and we run an experiment first without ozone and then with ozone. And we can vary the temperature just to see what sort of effect that brings, okay? With an idea for later recommending perhaps optimum ways of running your home, at what temperature, what relative humidity, etc. So we can vary the temperature, the humidity, the clothing, either shorts and t-shirt or long pants and long long sleeves. We changed the age. We had teenagers up to elderly folks in there, and we changed the ozone level, which is rather important. Okay, so here's some just data to give you an impression of how the experiment was run. And I have to say the data is still warm on the screen. We only finished the experiment 3 months ago, so everything I'm showing you here is kind of like a quick look direct from the measurement. And this is very much a typical experiment that's been done before. I think Richard alluded to this before. So this is, you bring the students in the morning, and they play with their phones for a while. And then in the afternoon, you put them back in, but put ozone on. And here I'm just showing you some of those classic ozone products which were mentioned before. The moment the ozone goes in, you can see that the products and the products of the products start to rise. And so we have data from the ozone side, but actually not shown here is a lot of the carbonyls, the alkenes, the acids, which we're normally emitting all the time, but they were also there. That's just to show you how the sequence works. So first without ozone, and then with ozone. And one interesting thing that cropped up was when we looked at the ultrafine particles. This was measured by a group based in the University of Lausanne in Switzerland. And they measured ultrafine, so we're talking like 1 to 2 nanometer size particles here. And under normal conditions, there was no real production of ultrafines. But the moment that the ozone was switched on, then suddenly we start to get ultrafines being produced around us, and they're growing in size up to sort of 2 nanometers. And that growth rate is strongly dependent on whether you're wearing t-shirts or shorts. Okay? So if you want to remember that, going home, think about Pigpen, the friend of Charlie Brown, as walking around with a cloud of particles around it, but you're generating particles around you all the time. Now, I talked about OH reactivity, and this is a technique This is a widely used atmospheric technique for outdoor chemistry, and we use it currently in our studies in the Amazon jungle. We do a lot of work currently deep in the Amazon jungle. And what we've done is we've developed a measurement method by which we can measure directly the total reactivity of the air. So in one direct measurement, we get a number for how reactive to the OH radical the ambient air is, and we get a number for that. And this is very handy because it allows you to do a budget and to find out how well you're doing at quantifying all the organics in the air. You can take that total number and you can compare it to the sum of the reactivities that you get when you sum up all the individual measurements that you've made times by their rate coefficient. And what you get is kind of like a budget that I've shown here as a pie chart. So you can say, okay, there was so much reactivity from all the measurables, isoprene, methanol, terpenes, etc. There was so much reactivity from the NO, ammonia, methane. But there can be a missing fraction. That was what we were after, to see if there was a missing fraction. And also to find out whether breath or skin was causing more reactivity indoors than between those two sources. So here's some sort of, again, rough data. This is not final correction, but it gives you the impression, I think, the first impression of the data. You can see in the morning, so the early morning section, where we've got something like reactivities measured in per second, we've got something like 10 to 12 per second. In that period, there was no ozone entering the room, and most of that reactivity can be attributed to breath-borne compounds like isoprene, which is very reactive. Okay? But the moment you switch on the ozone, the reactivity leaps up factor of 3, and suddenly the air is becoming much more reactive to the OH radical. And it's also producing a number of compounds which are harmful and inflammatory to your lungs. So the message here is, with ozone, most of the reactivity is coming from your skin, ozone reacting with squalling and setting free very reactive compounds. But when ozone's not present, then most of the reactivity comes from your breath. And indeed, it makes a big difference to, in terms of temperature and relative humidity and clothing level. Let's look at the carbon dioxide and the ammonia traces here. This is, again, just taken straight from the field, these plots. And what you see with the CO2 is that quite rapidly, after the people enter the chamber, the CO2 sort of rises to a rough equilibrium. Okay? Now, the peaks that you're seeing there, they're deliberate. We actually encourage the students to occasionally stand up and jump around, just to move around, not get too bored, and see what happens. So those are those sort of spikes that you're seeing. And you see them in CO2, of course, because the breathing rate goes up, and also, to some extent, also in the ammonia, because the sweating rate goes up. But while CO2 goes to the walls, goes to equilibrium relatively quickly, ammonia seems to be just carrying on, adhering to the walls. So what that's telling you is that you, in the room, we are now all sending ammonia to all the surfaces in the room. Okay? And on those surfaces, of course, it can undergo interesting surface chemistries, which will hopefully be investigated by other people in this consortium. It makes a massive difference if you run your house at high temperature and you walk around in shorts and temperatures. It's like a factor of 10 more in the ammonia flux if you walk around like that in a hot house than if you walk in a cold house with long sleeves and long pants. And I should add maybe here as well, we also have another group that's actually taking measurements of the chemicals which are in those clothes at the end of the experiment. So there's sections of the clothing which have been worn by the students was sent also to analysis. So we not only measure the gas phase products, but also the products which end up finally in your clothes. Okay, so this is basically the take-home message I want to tell you, is that you have around you your own personal atmosphere, and you're emitting into it hundreds of volatile organic compounds through your breath and your skin. And when those volatile organic compounds interact with oxidants, be it OH radicals or ozone, they produce hundreds and thousands of more of these compounds. On top of that, we're strong emitters of ammonia that can go to the surfaces. They may interact with surface reactions. They may feed microbes on the surfaces. We emit a lot of CO2, 4% in our breath. Methane was also a strong emission, and interestingly enough, old people emit more methane than younger people. So that was there's a greenhouse effect for elderly people, maybe. The particle story is very interesting. We're looking at that very closely now to see if we can model, see if we can explain how we can produce these fine particles in the face of the large particles that are present. And of course, when these reactions are going on, you're generating a whole suite of radicals, Kriege radicals, OH radicals. And we've been seeing also in collaboration with Alan's group, I'm looking for Alan now, there he is, we've been seeing some very interesting effects between radical product abundances that are affected by relative humidity. And finally, I mentioned microbes because we're also a source of microbes, and the next speaker is going to tell us all about that. But the final message, the sort of take-home message I want to leave you with, is that we are observing very strong effects in terms of the emission rate of the volatile organic compounds, ammonia in particular. And so that means there must be optima that can be found in how you live your life to minimize your exposure. And that optima may change according to where you live. If you live in a hot, humid environment like Florida, or a dry, cold like Alaska. So there's an optimum to be found. And with that, hopefully I'm on my time. I just wanted to say thank you to the people who did all the measurements, particularly Pavel Wagoki and Gabriel Becker, who ran the chamber. Sarka's been doing the clothes stuff. Dusan's been doing the particles. Our team did the gas phase organics, and we've been having huge help from Charlie and Glenn throughout the experiment, and a very big thanks from the matchmaking of Paula and the Sloan Foundation for funding us. Thank you very much.
Our next presenter is Dr. Rachel Adams, who will be speaking on environmental microbes and ambient moisture, how microbes contribute to the chemistry of our homes.
Thank you, Dustin, and thanks for inviting me to speak with you guys. So what I'm going to be talking about today are not the microbes on our skin, actually, but environmental microbes, so those naturally found in household dust, and how those can interact with ambient moisture in the form of relative humidity in the air to contribute to the chemistry of our homes. And we're focusing specifically on volatile organic compounds, or VOCs, so those compounds that become airborne. And here I'm showing a pie chart of potential sources of VOCs in homes. And we're specifically focusing on the piece about environmental microbes, both in a qualitative and quantitative sense. So how big is that slice of the pie, and what are the specific contributions? Now, I'm presenting these sources as static when all of these are actually dynamic. So all of these have sort of background emission sources interrupted with periodic spikes or enhancements, as we heard about earlier. Environmental microbes are going to be no exception to that dynamic. And second thing to point out is many of VOCs that are microbial are not unique. So there's microbial and non-microbial sources for VOCs. And a big challenge in this work is disentangling the two and isolating the microbial source for many of these VOCs. So microbes contribute to the environmental quality of buildings, both as a direct source of chemicals. As part of their normal metabolic processes, they will emit chemical products, but also in terms of transforming chemicals in their environment. So for example, microbes can actually or play a role in the degradation of plasticizers in household dust. And these chemicals not only affect our perception of that space and that building, but it has important implications for our exposures. So the one factor that's preventing all of our homes or all of our buildings from looking like this is water. So how much water is available? And water availability water can be available in buildings for a variety of reasons. They can be from floods, or from a building failure, generic term for something like a plumbing leak. But it can also be from relative humidity, so water that's just in the air. And ultimately, what we're trying to understand is what these environmental microbes do with the water that is available to them. So our previous work in this area began on surfaces that were intended to be wet, so kitchen sinks and shower stalls. And on the left, we have a microscope image of one of these coupons that was left in a kitchen sink. And you can see these coupons get coated in a film. And when you zoom in on these films, this ribbon of film ends up, is sort of revealed to be a ribbon of bacillus-shaped bacteria that has grown up on these stainless steel coupons. And we showed that there's a relationship between the total VOCs that are coming off of these coupons and the biomass of the microbes that are on those surfaces, suggesting a link between microbial production of VOCs in your home. But most of our homes are not designed to be wet. So what we wanted to do is move to areas that are not intentionally wet. And we wanted to build a framework for the relative humidity conditions under which microbes are active and contribute to volatile chemistry. And I'll talk about two aspects of that work that's ongoing. One is to identify the critical relative humidity levels that support microbial growth. So this is a pretty basic question, but one that has important implications for building recommendations and building design. And the second agenda is to use the time of wetness model to really try to understand and isolate microbial and non-microbial contributions to volatile chemistry. So this research is enabled by using a combination of innovative techniques in both chemistry and biology. So we're using an instrument that Alan mentioned that can detect these organic compounds, some in the parts per trillion range, in real time. So offering us really finely resolved data. And we can combine this with biological tools. So we can not only look at the composition and the species of taxa that are there and their biomass using DNA-based techniques, but we can also look at specific gene profiles that are being expressed through metatranscriptomics. So I'll give a brief overview of what we're doing with this work. Is we have two sample types that we're working with, carpets and drywalls. And we autoclave sterilize them, so we subject them to a high pressure oven to try to eliminate any sort of background microbes that may be there. And then we subject them to treatment. In carpets, we embed them with natural household dust. We also embed it with dust that's been irradiated to sort of maintain the dust matrix but eliminate the microbes. And we also have carpet only. And we're looking at dust from 3 regions of the US and from 2 different carpet types. For drywall, similar thing, we inoculate naturally in homes, compare that to drywall without that have not been embedded, and compare. And again, we're looking at different regions of the US and in different paint types. So first, to show what we're finding in terms of microbial growth. So what we're looking at is the amount of fungal biomass on these samples as a function of the relative humidity that they were left to incubate. And what you can see is that in carpet, you see a large flux of microbes that are fungi that are proliferating when the relative humidity increases from 75% to 80%. And in drywall, you don't see the same sort of growth until 95% relative humidity. Now, I should point out that we make all our comparisons with sterilized samples because we want to disentangle this microbial and non-microbial contribution. But it turns out that the techniques that we're using reduce but did not entirely eliminate all microbial activity. So I'm just showing as a comparison what the fungal biomass was on samples that were completely sterile. And so they were quite marginal compared to when microbes were there, but they were still present. So when microbes were there, at high relative humidity, you saw something like 30 million spores per milligram of dust. and our sterilized samples are closer to 20,000. So we're seeing a drop, but it's not completely eliminated. And this bore out when we actually looked at the material. So we, as I mentioned, we irradiated dust to try to eliminate the microbes that were present. And so we sent this to a company that kills things, usually on medical equipment, using gamma radiation. And so this was our sort of sterile dust. And then after we started getting these findings about maybe these, our sterile samples were not so sterile, we looked at what would happen when we grow these on a plate with food. And the top is your normal household dust. You see quite a rich flora. But on the bottom, we're looking at irradiated dust, and you can see it's not completely sterile. So what this shows is that dust is offering a really protective matrix for these microbes to persist, even under extreme circumstances. So next, moving on to the time of wetness model. And this is the model, or the idea, that microbes, or that environmental conditions in homes are going to fluctuate between those that are favorable for growth and those that aren't. And this is specifically in the context, again, of water, of how much water is available to them. And so we cycle these samples through periods of high and low relative humidity, where high relative humidity is 85 and low is 50. And the relative humidity of 85 is quite high, but not completely unrealistic in carpets, where it can also be high. Carpet relative humidity is often higher than ambient air relative humidity, or for example, after a shower. So these are periods of high relative humidity, but realistic. And in each of these plots, the low relative humidity are in gray and the high relative humidity is high. And I want to point out sort of how or not these compounds respond to cycling relative humidity and how that may indicate a microbial or non-microbial source. So here we're looking at benzaldehyde, and on the left, the concentration of this compound is not responding to relative humidity. However, on painted drywall, it is. And you can see that the dark blue lines have microbial inoculum on them, and the light blue line does not. And so this sort of difference between the substrate background and those with microbes we're attributing we hypothesize are coming from the microbes, actually being generated by the microorganisms themselves when they're growing and proliferating under high relative humidity. So another compound is a C8 unsaturated alcohol that's commonly called the mushroom alcohol because it's produced by a lot of fungi. And in both carpet and drywall, it drops in the case of carpet, it drops to background when the relative humidity drops and responds, increases when the relative humidity increases. Same with painted drywall. And it's one thing to point out is that there's a lag in the response. So the concentration doesn't increase immediately when the relative humidity increases, which is entirely consistent with a biological origin for this compound, because we know that from a quiescent state, microbes there's a little lag in how quickly they can sort of rebound and grow. So in addition to fungal markers or fungal VOCs, we also see indication of bacterial activity. So hydroxyphenyl acetate was a compound that was highly correlated with microbial biomass in our previous work. It's part of the styrene degradation pathway. We did not observe this in carpet, but again, we observed it under periods of high relative humidity in drywall. So this work is ongoing, but to sort of show up where we would like to take this. So this dynamic processes of looking at how microbes and surfaces respond to varying relative humidity is really powerful as a way to disentangle the microbial and non-microbial source for many of these VOCs. And the next step is to link specific gene compositions and gene profiles with specific VOCs. And I think this is going to be powerful because it's going to allow us to not only isolate particular bacteria or sort of biological origins that are related to chemicals of interest, but maybe also flag some chemicals that are tracers of biological activity indoors. And I mean, I'll leave with the point that we've known for thousands of years that living in a damp and moldy building is bad for your health, and we still don't know why. And it's a really challenging problem. I don't want to minimize that, but I think these tools offer a really powerful approach forward to tackling that long-standing problem. So I'll thank the Sloan Foundation for funding this collaboration, which is the group I'll show here and there.
Our next presenter is Dr. Barbara Turpin, who will be speaking on water, chemistry, and exposure.
Thank you. So I became an atmospheric chemist when I moved from the mountains of Washington State to Southern California to go to college. And my college is 5 miles from the San Gabriel Mountains, and I could not see the mountains until November when it rained. When I went running, my lungs hurt and my eyes watered when I went to class. And so I started working in Glenn Cass's lab doing atmospheric chemistry, and I gained considerable appreciation for the fact that air pollution is dramatically transformed by chemistry, and what you're exposed to is very different from what is coming out of the emission sources. Understanding chemistry has been really important to improving outdoor air quality. It's enabled us to develop cost-effective techniques to improve air quality. To improve air quality despite the fact that there are a lot more people and there's a lot more activity. Well, EPA is responsible for keeping our outdoor air quality healthy, but I got to work first in the indoor environment because the indoor environment is the major location where people are actually exposed to outdoor air. And so it was really important to understand how that outdoor air pollution is modified by outdoor to indoor transport. And so in the course of doing this RIOPA study, I learned quite a bit about indoor air quality and how interesting it is and how different it is from outdoor air. And so this just shows, for example, that the particulate matter, fine particulate matter that comes in from outdoors looks very different in composition to that emitted from indoor sources, emitted or formed indoors. One of the main contributions that I've made, I think, in outdoor atmospheric chemistry is the work I've done, along with others, to understand the role of liquid water in outdoor air chemistry. So it turns out that water-soluble gases in outdoor air are taken up by water in clouds and fogs and water in fine particles. And they undergo chemistry in that environment. When the cloud droplets evaporate, it changes the composition of both the gas phase and the particles. And quite a while ago, I guess Bill Nazaroff heard me talking about this at the AAAR conference, and he came over and he said, "Did you know that dampness in homes is associated with adverse health effects?" And you know, certainly at least part of that is microbial, but we really haven't been able to explain a lot of that effect. So he wondered, he hypothesized that perhaps water chemistry indoors might play a role in that kind of effect. And we don't know the answer to that question. But through the funding from the Sloan Foundation, my group has been able to start to look at some of the questions that relate to that one. And so the first thing we did was to go to 13 different homes in North Carolina and New Jersey and collect the total water-soluble organic gases in indoors and outdoors. And so what we learned is that the concentrations of total water-soluble organic gases are about 15 times higher here in red indoors than they are just outside of those homes. So we wanted to know, what are these compounds? And about half of the mass that we found here in blue and red is organic acids. Most of that is acetic acid here in blue. And so you see all the homes have something in common. They all have these organic acids present. And then I'm going to show you in the next couple of slides what we've learned about what's in the rest of the bar, the gray part here. And the remaining mass, that gray part, is really complex. They're just a very large number of compounds, and these are some of them. There are compounds that are commonly used as solvents, compounds that you do find from microbial sources, disinfection byproducts, and some that you find emitted from pesticides and cooking food sources. And one thing that we found is for this other half of the water-soluble organic compounds, there's a lot of diversity from home to home in what you see. So each column here is a different home, and each row is a different compound, and if you see a white bar, it means that compound is present. So there are a few compounds in this mix that are present in all the homes, but a lot of the compounds are present in some homes. You know, there's a lot of variability from home to home. If you have real-time measurements are really useful because you can learn a lot about the sources and the sinks and the chemistry by looking at real time. So anyway, we took this chemical ionization mass spec to one home. We benchmarked this home against the other 13, so in many ways this home fits within the same, it's very much like the other homes in many ways. It's a 15-year-old home with forced air heating and air conditioning. We made these measurements in the summertime, and so the heater wasn't on, but the air conditioner cycled on and off. And so this is one of the things we found. While the temperature in the room and the relative humidity in the room were really quite well controlled and very constant, fairly constant throughout the time, we measured the relative humidity here, the dotted line, and the temperature at the air supply vent. So this is where when the air conditioner kicks on, you feel this cold air coming out of the air supply vent, and that's where those measurements are made. And then here in blue is the acetic acid concentration in the home, the formic acid and lactic acid. And in gray, you can see that they go way down when we sample from outside instead of inside. And so every time that the air conditioner kicked on, the acetic acid and formic acid concentrations dropped. And so what's happening is that acetic acid and formic acid are being lost in the air conditioning system. We suspect they're being lost to the wet surfaces in the AC duct, and also the air conditioning coil is always quite wet. You can see this same pattern for many different organic compounds here. These are compounds that you find in solvents, plasticizers, cosmetics, perfumes, food and flavorings, and additives. And so all of these compounds show a cycling with the AC system. So this suggests to us that highly likely that these water-soluble compounds are being taken up by the wet surfaces in the AC system. It's very possible, although we can't say for sure, that there is chemistry that happens in those wet surfaces. And it's also possible that those water-soluble compounds, or some of them, could be a source of nutrients for microbes on the AC coil. In order to explore in a more controlled environment, we built this flow reactor. And so this is a parallel plate flow reactor that allows us to put real indoor surfaces in here. We can pass a single water-soluble gas through this reactor, and then we can move this the sampling probe up and back in order to measure the remaining concentration of the compound. So we've done experiments with clean glass and clean painted drywall, and then we have glass and painted drywall mounted in people's homes so that they can they can soil naturally in those environments, and we can do controlled experiments with realistic, authentic indoor surfaces. And so, for example, here, this is acetic acid, and its uptake, this is the concentration here of the acetic acid going into the reactor before it's exposed, and then you can see that the concentration goes down as a function of distance through the reactor because the acetic acid is being taken up by the clean painted drywall. And that the uptake of that acetic acid increases with relative humidity. And so that's pretty good evidence that this water-soluble gas likes to be taken up into these real materials. Okay. So it's plausible that indoor water will alter indoor exposures. We know that uptake and reaction on wet surfaces is important. It's quite important outdoors. And there's at least 300 times greater surface to volume ratios indoors than there is outdoors. Even if you have simple water adsorption, that means at least 20 times the water per volume as you have outdoors. We know the concentrations of water-soluble organic gases are higher indoors than they are outdoors, and that about 20 to 50% of homes are considered damp. And so this could have implications to inhalation and dermal exposure if the products of water chemistry, if water chemistry leads to volatile products, then you inhale those, right? If it leads to non-volatile products, then that's a sink for those compounds, unless, of course, that chemistry happens on your skin, and then you're dermally exposed to those products. So there's a lot that we don't know, and I think that the role of chemistry in moisture-related, damp home-related health effects is still a good question. It's an active hypothesis. And so I'd like to leave it there and acknowledge the funding for my current event, which is the Sloan Foundation, and the Health Effects Institute funded our original indoor work. Thank you.
Well, thank you. We'll start with questions. And for those who are watching via the webcast, you can submit questions online in the chat box or by Twitter using the hash string that was available. I'll start off with a quick question just to get started. A lot of the discussion was on the indoor environment and how microbials or even water chemistry can impact the indoor environment. One of the biggest tools that we have for controlling the indoor environment is our heating, ventilating, air conditioning systems. Have there been any discussions or studies looking at how we can improve how we design and operate our HVAC system so that we can make better improvements to the indoor air quality, or is that ongoing or future research that's to happen?
Well, there has been work looking at specifically air conditioning, cooling, cooling coils, and specifically the potential role that those play as a source of microbes and microbial VOCs into homes, and different ways of potentially sterilizing the HVOC. But maybe other people would know a little bit more about specific HVAC work. I don't know it.
I think generally houses are designed around energy mostly to make a comfortable living space, and it's these new discoveries which challenge us now to rethink how we want to operate and build our homes to create a healthier environment. The fact is we didn't know about most of these emissions or a lot of these emissions before, and so the original way you build a house was supposed to be to keep yourself warm and preserve as much energy as you can. But knowing now what we've seen here just now, there may be strategies that you can adopt to minimize your exposure, simple ones too.
Any questions? Any additional questions?
Go to the mic.
Could you please,
This is a question for Dr. Turpin. Do you think the solubility of these gases in water differs from the bulk measurements to droplets? Have you ever tried to investigate that?
Mm-hmm. Yeah, this is a really interesting question. It certainly does. In outdoor air, we know that the uptake of these compounds to cloud droplets, which are really dilute, is very different from the uptake into aerosols, where there's more stuff than there is water in each particle, right? And so the uptake can, in some cases, be quite enhanced to the aerosol, and the chemistry is different. So we expect the chemistry in wet drywall, for example, to be very different from the chemistry on your AC coil.
So just to follow up on that, it sounds like what you're saying is that atmospheric chemistry, we're dealing with a real water phase, but down near the Earth, we're dealing with a hydrated phase.
Yeah, I think that's right.
Okay.
any questions? I have a question online for Dr. Turpin. Have you looked for perfluorinated acids in the aerosols?
Oh, yes, so this is a question about PFAS.
Yes.
Right. Actually, we're involved, and Heather Stapleton too, here in a big PFAS project in North Carolina. The state of North Carolina is funding, and all of that work really is driven by concerns over health, most of it about drinking water, contaminated drinking water. But we know that wet deposition of PFAS in North Carolina is an issue, that rainwater is contaminated with PFAS. And my group is working on measuring particulate and gas phase PFAS outdoors because the state is worrying about health. But I think that we really expect and know from the few measurements that are available in other places that indoor concentrations of gas, of PFAS in the air are going to be higher than outdoor concentrations. Maybe if you're in the plume of the Chemours faculty facility, that's different. But so we have measured PFAS in ambient particles outdoors, and we're really looking for the opportunity to do that indoors as well.
I have another online question. It states Dr. Williams talked about quantifying reactivity in terms of OH chemistry. The question's for all the panelists. Have any of the panelists seen the importance of chlorine chemistry indoors? And specifically for Dr. Williams, how would that affect the OH equation?
Oh, that's a good question. Well, I think there's been some nice studies that have been presented at previous meetings from the home care team. Do you want to say something about it, showing chlorine chemistry in action? Do you want to say more? I'm not sure if your mic is on. Ah, okay. Sorry. Okay, is that better? Yeah. So I understand that the home care team have actually done that experiment by generating chlorine radicals through the use of bleach on floors. Do you want to say something about what you saw?
I was not involved in that.
Oh, I'm sorry.
I don't know.
Ah,
You'd have to ask Nina.
To get the home chem, do you want to?
The question, I was too busy tweeting, sorry.
okay.
About chlorine radical chemistry indoors.
Yeah.
What do we know about it?
Have you seen it?
So we see that chlorine used during bleach cleaning, we mop the house with bleach. We observe that there is interesting chemistry that's going on, both in terms of inorganic and organic chlorine reaction products. We did observe formation of secondary organic aerosol that organic. We don't really know if the aerosol is organic or inorganic at this point. So it's I think it's organic, yeah. But, so we're still digging through the data, and I don't have published results, but what we know is there's substantial change both in the surface and the air chemistry from bleach cleaning.
There's one more.
Oh, that's all right.
I have a question for Dr. Williams. In your studies in the chamber in Denmark, you noted that methane production was higher for older people than for younger people. Did you do any studies on the basis of diet variations for some of the students or subjects?
No, that's an excellent point there. That's something that we would like to do in a future phase to see whether the effect of diet changes your general emission pattern through the skin or through the breath. It's a very good point.
Any other questions? Kim?
So back on the methane or other questions, the issue is in what you've done in your control studies, have you picked any subjects who were, for example, obese versus lean, that kind of thing? Because I would think that our internal microbiome is going to make a huge difference.
Yeah, indeed. That's another good question. In this particular study, we tried to standardize as much as possible. So we tried to use, for most of the experiments, sort of basically student-age people from 20 to 30 with a certain BMI that was reasonably healthy. But indeed, that's a good, there's so many variables that you can do. Once you've got this setup, the chamber setup with everything under control, I showed you some variables there with ozone, temperature, humidity, there's sort of basic set of variables, but there's of course a huge number of other ones. And indeed, yeah, that would be a good one. I mean, depending on how metabolically you burn fats, you produce very different molecules on your breath. And that could also impact. That's a very good point. Yeah, if you have any other suggestions for variables that we should vary in the future, then don't be shy. Tell us in the coffee break. Yeah, we're very keen.
Most of the examples we've seen, the experiments have involved modern houses with modern HVAC. I'm old enough to have grown up in a house with steam heat and whistlers, and I recall water formation underneath the little whistling elements. How safe were old houses with steam heat, given what we know now about this subject?
Well, you're alive, so.
I would say that from what I've heard, it wasn't a very safe house, but everybody in that house lived the older people lived to their 90s, and the younger people were all still alive in their 70s and 80s. So I'm wondering. You know,
it's an interesting thing that we've had many discussions about old versus new houses, just, out in the coffee break area, that kind of conversation. The RIOPA study, because we looked at homes in Elizabeth, New Jersey, Houston, Texas, and Los Angeles County, California, it was really interesting because, in part because of the home age and the home construction and the way, and the climate that you're operating your home in was so different. In Elizabeth, those are 100-year-old homes, and when it gets hot in the summer, people open their windows. And in Houston, there just aren't very many houses, even in the shipping canal area, right? There are some, but there aren't that many houses without air conditioning. So, but if you think about the health risks of old houses versus new houses, there are health risks in both cases. And to me, and I've lived in now a 100-year-old house and a 20-year-old house recently, but to me, it seems like we know a lot more about the health risks of those 100-year-old houses, right? There are things like asbestos and lead and radon, perhaps, in both cases. Just sort of as a citizen who spends too much time indoors, the way I feel about it is the new homes, there are so many new materials made with new chemicals, and there are new chemicals used, more and more new chemicals used every year in consumer products and in materials and furnishings that we really don't know very much about them. EPA has a database of chemicals in consumer products. They've got a list of 75,000 chemicals in 15,000 consumer products. And I think we have good toxicity data. Maybe Linda knows a better number than I do, but about 200 of those. And we really don't have very much data on what's in the ambient air or what people are exposed to. Most of those, we just don't know very much about them. So, I don't know what I can tell you about which of those risks you prefer, but the old homes are really different from the new homes.
Yes. Well, thank you, everyone, and thank you to the panelists. This ends this panel, and we will be... Some announcements. Okay.